continue with Yallapu sir's second lecture, FDA Approved Nanoformulation for Clinical Application.
Welcome back again. So I think, just now we learned a lot more about nanofibers, okay, which I have not at all covered here, okay. So when I was deriving, so here, we always, focused more on injectables, okay. That's why we didn't cover a lot more nanofiber work. And also, like, we do have a dedicated topic seminar, I think tomorrow, on preparing various type of nanofibers and its application in various disciplines. So as I mentioned before, like whenever we talk about nano material, okay, according to FDA of United States or national, I think, NIST of USA, they define that nanomaterial could be any of dimension of a particular size in nanometer size range from 1 to 100 nanometer. Okay, it could be any morphology, size, in the dimension range. It could be a fiber, it could be a particle, it could be a rod shape or anything, triangle nanoparticles, star shaped nanoparticles, nanofibers, and so on. Okay, but since our goal was more of injectables rather than surface attachments, like wound healing or some of these regenerative things that does not require type of injectables. Okay, you can make hydrogel based, okay, kind of bulk hydrogel based, but you embedded with the nanofibers. Any of that kind would be sufficient. But here we are dealing more of like injectables that it can go into the body. So this is the one slide is dealing with and telling us what type of nanomaterials has been exploited for various medical applications. Okay, if you see this slide, we have 4 type of nanomaterials that has been widely applied for medical applications. Okay, so number 1 is liposomal based or lipid based nanoparticles, and the next one is polymeric micelles, third one is polymeric nanoparticles, and fourth one is metal based nanoparticles. If you see on your left hand side, the requirements, like if you want to have a targeting of tumor cells, or to want to target the vasculature, or if you want to have a trigger based drug delivery, means like pH sensitivity or temperature sensitivity, or redox based, like so at the tumor site there are more glutathione. Okay, so you make a nanoparticle such a way that when it goes and see the glutathione, or in the presence of glutathione, the polymer networks will be dissociated or chopped out, and then drug will be released. Okay, so based on the those type of requirements, and also passive delivery or the increased stability, slower elimination, if you see these type of requirements, the liposomal formulation, okay, one way or other way, it has been satisfying all those properties. Okay, from top to bottom, everything is liposomal formulation or lipid particle formulation is obeying. Okay, so that means it's already proven. So therefore, even the mRNA application was way easier when they want to convert that liposomal based formulation for delivering these therapeutic mRNA into these nanoparticles, and then they make effective formulation of those vaccines. Polymeric micelle, to some extent, it's not obeying. It's not possible that targeting vasculature, targeting tumor cells, because as I said before, because of it doesn't. Whenever you put in the human, it's all the polymeric chains will be like dissociate from the polymeric micelle structure. Okay, because of a grade variation in the concentration. And same way, like polymeric nanoparticle, a little better than your polymeric micelles. Okay, and third one is metal-based. Mostly can be used by trigger-based, passive, and some of the other. Okay, the overall goal is that we have to think that what type of nanoparticle formulation that we can make that has all type of like these characteristic properties that we can achieve. Okay, so with that, I would be stopping here, but just I want to show the journey of the various type of nanoparticles, how the field has been evolved right from 1964. So you can again recollect that liposomal formulation was the earliest one people has been using for various applications. Then you keep going, and then now you see novel-based formulations like cellular membranes coated nanoparticles and so on. Okay, but anyway, we'll be covering most of these formulation in our FDA approved nanoparticle formulation. Before I go, just we want to introduce our own work, just to have some feeling that you see what type of work we do at UTRGV and our previous institutions. So we heavily focused on PLGA nanoparticles and magnetic-based nanoformulations. So here one of our work has been heavily put emphasis, constructed iron oxide core with the different layers. See that different layers, and then you put them drug molecules, and so on, curcumin and everything, and we have a patent on this area. And another one example is the cyclodextrin-based formulation with the curcumin. And the same thing, I talked about PLGA nanoparticle. I showed that it is better than plain curcumin, and here we explored that how much better if we make PLGA based curcumin nanoparticle formulation. We tested right from cell line models to animal models, and also we tagged with the PSMA antibody that is particularly recognize the prostate cancer cells, because those cells have almost all prostate cancer cells, they express PSMA protein on those cells. So this is one of the best target. And you can also remember that PSA based imaging technology is already approved by FDA. So means that they take PSMA antibody and they conjugate with the either radioactive molecules or any fluorescence tag. Okay, when they inject, and then this will go and bind to the tumor cells wherever in the body. Okay, so you all need to remember that whenever we talk about cancer, it's a originated cancer from a particular organ. Okay, for example, breast cancer originate in the breast, prostate cancer originate in the breast, lung cancer originate in the breast. Lung. But at the same time, these cancers can metastasize to various other organs. Okay, for example, if you take breast cancer can go to the bone, breast cancer can go to the brain, breast cancer can also go to few other organs. Okay, so whenever we talk about imaging technology or therapeutic technology, let's say, you keep in mind that you want to deliver to the prostate cancer means not necessarily just only to the prostate. Okay, earlier time it is fine, but once it is metastasized to the other organs means like you need to have a better targeted moiety so that that targeted moiety can track the cancer cells wherever those are, then go and bind and it's effectively work on those particular cells. And we repeated the iron oxide based formulation with the known chemotherapeutic agents. We tried earlier with the nutraceutical agent called like curcumin and few other. So here we were testing whether the same technology or same nanoparticle can be applied to the existing chemotherapy. Those were approved by FDA. So again here same nanotechnology, magnetic nanoparticle formulation with the same layers, but we here replaced with the microRNAs. And also we can replace with the siRNAs. Okay? So we found some of the microRNAs are lost in the case of some of the cancers. Okay? So that means if you deliver or if you supplement that particular microRNA, maybe it will restore its cells to cancer to going back to the normal cells. Okay? So as I mentioned earlier, like here the field evolved, and then you see here cell membrane coated nanoparticle evolved like a different formulation. Okay, so that means it has its uniqueness, that we can take advantage of it. So what we did here, so here we made some of the nanoparticles as a core. Okay, here PVP nanoparticles, but on the PVP we put a natural molecule that is polyphenol called tannic acid. Okay, you have that tannic acid layer that you can put whatever you want. You can put drug molecule, you can put any peptides, proteins, whatever we want. But to be as a targeted delivery purpose, we here used 5 different type of cell membranes. Okay, the cells like we used neutrophils, natural killer cells, mesenchymal cells, and then Jurkat cells and raw cells. Okay, these are all we got those cells, and then we extracted membranes of those cells, and then we put it on these nanoparticles. Okay, so we coated with those cell membranes, and then see how better that we can target tumors on the mice. Okay, and finally we end up having more targeted data. Then we identified that neutrophil based coated nanoparticles are much better than any other coated nanoparticles. Therefore we used in the animal model for therapeutic application. So another one is that this was a PhD student. She graduated in 2021. This was a postdoctoral fellow work. What he did, so as I mentioned before, we have heavily focused on polyphenols like curcumin, tannic acid, resveratrol, all those. So here, how about we use tannic acid itself as a delivery vehicle? Means tannic acid based nanoparticles. Okay, here is the one of the example that we did. So we cross-linked the tannic acid nanoparticles and we loaded a chemotherapeutic drug. Same way we can use whatever that Dr. Sabu Thomas mentioned, you can put titanium oxide, you can put zinc oxide, whatever we want we can put in these kind of systems. But only thing is that here we have a suspension based. Okay, so those were bulk based. Okay, so you draw a nanofiber that is physical, you cannot inject those. Okay, and we tested this better efficacy of those therapeutic agents right from mechanic aspect of those particular drug biologically, and also we test them in the animal model. So we know that if you use some of the polyphenols, okay, they have a good chemosensitization property for chemotherapeutics, and also they have a good radiation sensitization property. Okay, that means that if somebody is undergoing chemotherapy or radiation, if they consume curcumin or gambogic acid in this case, it sensitizes, or on the other hand, it reduces the required chemotherapy that patient requires. Same radiation, like so if somebody is getting 5 doses of radiation, if you take this gambogic acid based, it can be reduced the amount of dosage radiation. So here we are showing you that when we combine gambogic acid with a drug called gemcitabine. Okay, so gemcitabine is a first therapy line drug for lung cancer and pancreatic cancer. So we are testing to reduce the amount that requires gemcitabine for treating lung cancers. Okay. On the other hand, we can also think that the cancer cells, after 6 months of treatment, cancer cells develop resistant to that particular drug. Okay. The nanoparticle construct we use is that tannic acid based again. So why we are again and again using tannic acid means that's the among many polyphenol, this is the widely used and can be dispersed in the aqueous phase. If you take many other, they are not dispersible. Even the gambogic acid, although it's polyphenol, we won't be able to disperse in water. So that's why we are using tannic acid based platform and focusing how better we can cross-link or how better we can deliver things using. So we proved that here you see the tumor growth control nanoparticles alone, and here you see gemcitabine alone, gemcitabine plus gambogic acid alone, and here you see the combination through the nanoparticle delivery. So it's like here the tumors are negligible here, whereas controls are way high, and the combination also you see visible tumors. Okay, so there we concluded that this platform is good. And as I mentioned before, if you want to target any specific macrophages, okay, so we put some of the same again, our emphasis on tannic acid based. So we put mannose mannose receptors are highly expressed in macrophages, and especially M2 macrophages. So therefore, we put the mannose conjugated on this nanoparticle, and then we effectively delivered on M2 greater than M1 targeting here. Well, so if we have a tumors already metastasized to the bone, okay, so we need to have a formulation that can go more effectively to the bone, right? Here we have a generated or constructed again tannic acid based formulation, that coated with the zoledronic acid. So zoledronic acid is a FDA approved bone targeting agent. Okay, it's simple, you need to have some idea where you want to deliver and how you want to deliver. Okay, so we show that when you have this bone targeting agent, you see a lot more bone binding along with the tumor delivery compared to here you have very bone less targeting. Okay, so when you want to deliver to the brain, you need to have some formulation that. So we are generated PLGA based formulation coated with some of the pluronics, where you can see good co-localization of the nanoparticles of with the different ratios, and then you see good accumulation into the brain. These are oral formulations. Okay. So for cardiovascular application, we applied long time back one of the nanogel formulation. Okay. So here when you do the surgery, balloon injured surgery, okay, what happens? Here, you make that balloon injured surgery, and then so that your heart hole, okay, the heart, not the heart holes, the vessel taking to the heart, those were completely blocked. So you do the surgery so that it will be opened, but because of more proliferative activity at those areas, the smooth muscle cells, they regrow. Okay, so what we did, after done this balloon injured surgery, we use a nanogel formulation that contained rapamycin, where it can still have this, the place that where the blood can flow easily to the heart. Okay, not the regrowth of the smooth muscle cell in that area. Whereas in the control, you see that blockage of this overgrown of the smooth muscle cell. So these are some of the example, how we want to think and develop a nanoparticle formulation for a particular application. Okay, so these are some of the references, that what I gave some of the backgrounds, but whatever our work we shown here, you can go into my Google Scholar, and then you can see all of them, or if you need anything, I can send it to you. So any questions so far in this area?
Sir, I would like to ask on trigger-based nano delivery, particle delivery to the mRNA vaccine. Mm-hmm. So please elaborate on the methodology of that delivery system.
Yeah, so here when you talk about mRNA technology, their main concern was the stability, not the trigger-based. Okay, so trigger-based is required when you think that, if you want to minimize the toxicity onto other organs. Okay. So in the mRNA technology, their main opinion was that, they want to preserve the mRNA because that degrades very fast. Okay, that's why all Pfizer or Moderna vaccines, those were kept in -80. Okay, so whenever you go, they just pick from the -80, and then they quickly make those suspension, and then they will give us injection.
Both Moderna and
both Pfizer. Moderna and Pfizer. Okay, so whereas the Indian marketed one, they supply at room temperature, and also Johnson and Johnson, they have a room temperature, because those are not mRNA based; those are live attenuated based, different kind of vaccines.
Sir, you also spoke about the heart block.
Yes.
How the nanoparticle is delivered to the block area.
So those were intravenously injected.
Okay.
So that means those were going everywhere, not only into those organ. Okay. Wherever the blood flow is there, they keep going. But what we observe, not only through this paper, and our team further did more work in that area. Okay. And we this was the one I showed only how to make those nanoparticles, and then I showed only one example, how the blockage was taken care. Okay. But that work was published in Blood Circulation and Intervention from Cleveland Clinic.
Okay, sir,
but so that was intravenously injected.
Okay, sir. But about the chemotherapy radiation reduction, okay. How nanoparticles target to reduce radiation effect, the effect of radiation?
Effect of radiation, So what happens, like, sensitization can be, it comes from various forms. Okay. For example, like, if I take chemotherapy, gambogic acid. Okay. So whenever you see a gemcitabine delivered to the cancer cells, after a while, the function of the gemcitabine, how it works, like, different pathway. So that pathway no longer will be obeyed by the cells. So when we did this gambogic acid here, the pathway that what I was talking about, like, RRM2, so this is the main mechanism that is responsible for gemcitabine resistance. So if you see here, RRM2 in the control, it's more. Gambogic acid, it's a little bit reduced, but when you treat with the gemcitabine, it's even more compared to cancer cells alone. So when you combine with the gemcitabine plus gambogic acid, you see complete reduction in the RRM2. That's one of the mechanism. But we did lot more other that we are not. I didn't put the slide here. What we did a RNA-seq analysis of the cells treated and then given for all the proteins that were upregulated and downregulated. We have seen and published a paper, and I didn't put on this slide because I thought that this is mainly for nanotechnology, not the biological aspect.
The regulated and down.
Yeah, so all the drug-resistant proteins gone down with the combination compared to alone treatment. And here I said radiation sensitization, but we didn't do that work by ourselves. But there is a report with the gambogic acid sensitizes to radiation with the iodine 125. Okay. Slow doses, it sensitizes. But that's not our work. I didn't show it here. Yeah. Thank you.
Thank you, sir.
So any other questions? Okay. So if not, oh yes, yeah, yes. Yes. So I think all biodegradable polymers we can choose in the first place, and majority of the biodegradable among all of them, widely studied material is PLGA. PLGA, yeah. Okay. So I think here I want to educate you more regarding how these nanoparticles are helpful. Okay, and how and what basis the FDA was approved. These are all formulation. Okay, just I want to take you, just I want to show what the learning objectives would be for this topic is that we will learn that advantage of nanomedicine and why it is needed and the current status. And type of nanoparticles or nanomedicine that we commonly employ, and FDA approved formulation, and some of the clinically used nanoformulations. See again that I put the basic slide information, how we distinguish or differentiate various type of nanomaterials and nanoformulation based on its type of liposome. You see, it's a DL layer lamellar based formulation. Okay, if it's only one, polymeric micelle like this. Okay, so polymeric nanoparticles. So your drug is dispersed either in the core or into the other area of the nanoparticle. If you see magnetic nanoparticles, it could be anywhere in the nanoparticles. So here liposomal, you see some of them are in the core or some of them are in the middle layer. Okay. So here these tags or tails, those are hydrophobic in nature. That means if you want to have a hydrophobic, that will go here. And if you want to put the hydrophilic, that can go here. So this would be the advantage, is that you can choose, or you can put like both type of drug molecule in this type of nano system. So here again, you can choose like what type of nanomaterial and what type of drug you want to put it in. Okay. So polymeric micelles are same way. So this slide is giving us information so far. When we did this information, we had 51 FDA approved formulations, and now I think with the Moderna and everything, I think the number of formulations went up. But when we collected this data, so this is the FDA approved nanomedicine based on the micelles, metallic, polymeric, nanocrystal, liposomal, and polymeric materials. When you see the nanocrystals, you see large amount of research was done during 2001 and 2005. So that time it was a hot topic that nanocrystal itself, like you have a drug, you make a crystal out of it with some kind of stabilization. Okay. So, but with time that has been changed, and then no more nanocrystals have been used, because there is no targeting, or there is no other advantage that we get from these nanocrystals. Okay, so I don't want to read each and everything, but here just I want to focus some of this, 1957, you see this formulation called Dex iron. Okay, it's nothing but iron with the dextran. Okay, it's a iron oxide based formulation they have been using for a long time to improve the iron deficiency condition in the kidneys. Okay. On the other hand, you will be supplementing iron through nanoparticle formulation. Okay. It was 1957. You can see a lot more other examples, like here delivering some of the IL-2 through the nanoparticles, delivering some of the antiferrocin B through liposomal formulation, and so on. Okay, so this is not it's very tough that we cannot give more and more examples, but these are all approved by FDA based on various molecules or various agents that we can put it in the nano formulations or in the complexation or self-assembly process, so that we can improve its activity and site-specific action. Yeah, so this where we will focus a little bit more time. So how and what type of nanoparticles formulation previously used to approved by FDA? Okay, it's very simple. Okay, so take a liposome and then you put a drug into it. Okay, so that was the basic one, doxil. Okay, so they choose a liposome and then they put a doxorubicin into it. Another example is that Feridex IV. It's a iron oxide nanoparticle stabilized with the dextran. So that was the way back that simple idea, but it's very effective. Nowadays, what new formulations are coming up. Yeah, you see the antibody, fragmented antibody, single chain, double chain, decoy antibody, aptamer, and so on. They conjugate or decorate, and then they improve the site-specific delivery. Okay. And now we can go through one by one. Before we go to one by one, just I want to show one example. Aspirin, it is some kind of drug. It has been used for over 120 years and over. Okay, it's known as a agent for headache, like it's a anti-inflammatory agent that has been used. It's proven, but the only difference is it was created like 125 years ago, but with time the formulation has been changed. See, from 1887 and 1899 is approved, but you see the journey of the same drug, but they put it in a different way, and then finally now the currently we get the same drug but in a different form, that is the microparticles. Okay, it's simple. What they did, they put sodium carbonate based nanoparticles. They put this drug, and then once you take this pill, it is more active than its other type of formulation, because it goes to the inflammatory site more heavily based through these nanoparticles. And doxorubicin, as I mentioned before, why that is more effective than its parent molecule. So Doxil is a polyethylene glycol coated liposomal formulation. Always, whenever we say nanoparticle, again those nanoparticles will be surface modified or surface decorated. So here liposomal formulation, but it is coated with the polyethylene glycol. What's the advantage here is, so you will be removing the RES clearance mechanism of the drug when you use this Doxil formulation. And at the same time, you increase the time of the drug in the body. Means like, you will be increasing stability or, circulation of the same drug when you put them in the nanoparticles. Okay? So these are the two advantages, but it has been used for various indications, for example, like ovarian cancer and various other cancers, breast cancer and other, melanoma and so on. So what it does basically, the main disadvantage of this drug is that this drug goes, wherever the active proliferation is there, this drug goes into the nucleus, and then it kills that particular cells. And you see the most aggressively growing cells in the body is that it's a smooth muscle cells in the heart, right? So that means this drug will go more into the heart and then it harms, okay? So when you put this into the nano formulations, what it did, rather than directly going into the via blood to the heart, so it increased the circulation time. Means it is in the circulation, but it's not going into the heart. Okay? So that's where the cardiotoxicity is completely either absent or reduced. Okay? That was the purpose. You need to find out what the disadvantage with the drug is coming, and then how we can improve. So the same technology being applied for another drug called amphotericin. It's like an anti-fungal agent. Okay, the same technology, but you choose a different drug. So here is the another drug for eye applications. Age macular regeneration. Same formulation again, liposomal formulation, but the drug is different. So another interesting formulation is that Abraxane. Okay, Abraxane is simple thought, simple idea, nothing but what they did. The paclitaxel is a drug. Paclitaxel is a drug, it's heavily used for various cancer applications. But again, the problem is the paclitaxel goes into each and every cell, okay, and it binds to the tubulins. On the other hand, the earlier formulations, since the paclitaxel doesn't dissolve in aqueous phase, they used to use some of the dispersing agent that itself toxic. Okay, so it's a camphor-based formulation they used to use that is giving more side effects. Okay, to minimize that, paclitaxel bound protein nanoparticles. So they choose human serum albumin as a carrier, but conjugated with the paclitaxel, and then those for each HSA there are about 6 to 7 paclitaxels were bound, and then when you process them, each aggregate or each human serum bound PTX is formed to 4 to 14 nanometer, but overall formulation forms close to 120 nanometer overall. What it did, it minimized the use of other excipients that we used earlier to deliver this paclitaxel. Okay, so the need was to eliminate one of the ingredient of the paclitaxel formulation, but replace with the albumin-bound paclitaxel nanoparticle. On the other hand, you have an advantage of using HSA-based formulation because it can also act as not only the passive delivery because of the size range of 120 nanometer, but also you can take the advantage of the GP16 protein targeting with the HSA. Okay, so HSA binds automatically to the GP60 protein. Anyway, GP60 protein is overexpressed in various type of cancer cells. That's another advantage of using this. And this was completely replaced by earlier formulation Taxol with the Abraxane based. See here, gray bar, it used to be Taxol, okay, because of the hypersensitivity that is coming from the Taxol formulation. The new paclitaxel bound with the HSA, it's completely replaced. Okay, now you see nobody is using even in 2018. Now no Taxol exists basically in the market. So another formulation I was talking to you earlier on, PLGA based formulation, right? So it could be you have a PLGA formulation in the solution form, and then you put this acetate drug that works on prostate cancer cells. But this is a localized therapy, okay? So they take this and then they right into the prostate, they inject. The only advantage with this system is that the drug in the formulation, it will be released over a 6 months period. So that's why this therapy is once in 6 months. You inject and then it's slowly releasing, and then it does its activity, like administer and then it solidifies, and then systemic drug release over a period of 6 months. And as I said before, some of the iron oxide based nanoparticle formulation, they not only give contrast properties, but also they have a hyperthermia property of those nanoparticles. So that property has been explored by a German company. What they did, say if this is a tumor, and then you right here you inject these nanoparticles and then put it under the alternate current, and then the tumor cells will be cooked. Okay, the temperature goes up, and then the cancer cells will die. Okay, that's the mechanism that they applied, and the advantage of using this method is improve the survival of the patients. Okay, so whenever we talk about therapies, for cancer or any other disease, for cancer there is no cure basically, right? So you will be improving the survival time. Okay, if we know the breast cancer earlier on, we can get the patient. The primary therapy is the surgery. Okay, they remove the tumor part and then they give some chemo or radiation. Okay, that method, a 5-year survival is like 90% people live even after 5-year surgery. Okay, but some cancers, for example, brain tumors and the pancreatic tumors, so those people, they live once it diagnosed maximum 6 months or 1 year. Okay, that means whatever we are giving therapies, that only helping few months of survival. Okay, whatever therapy that we are talking about, it increases few more months or few more years, depending on the type of tumor they have. Okay, the only idea is that you see how much, like from 6.2 months to 13.4 months. So in this case, so 14.6 months to 23.2 months. Okay, same way, like whatever the doxil we talk about, like other drug formulation, again it works as a plain drug, but only the advantage is that either it reduces the systemic toxicity or improve its side of action. Okay, we are not creating anything new, but here we are facilitating the process of effective delivery of that drug. Okay, and few more other examples, like this TNF alpha is a tumor necrosis blocker. Okay, so the same formulation, like if you make this formulation with some of the stabilizing agents. Okay, this is antibody, but you stabilize more so that you can increase stability and then effective targeting of those Grande disease cells and so on. So here you see what you have, that antibody here, only the increased activity, like you put PEZ conjugated here. Okay, that's the only modification we did. So because of this, you see, like what we reduced here, but at the same time we increase the injection site reactions, but it is much better in the activity and so on, like And ferraheme is another iron oxide based formulation. The advantage was that when we talk about first Ferredex formulation, that was iron oxide nanoparticles, but decorated with the dextran. What happened that that was approved for kidney supplementation of more iron, but the same formulation was applied for liver imaging as a MRI contrast agent. Later on, that has been recalled. Okay, so I will be telling what was the reasons. But here, like new formulations of same iron oxide formulation, but instead of doing the dextran coating, here they use a different polyglucosorbitol based effective coating. Therefore, this has been replaced with that iron oxide based dextran coated nanoparticle formulation. These are all the different strategies that we commonly experience when you are making a formulation. This is the example of the Ferredex. So why it has been chosen as a contrast agent? Because it binds very well, macrophages, all those cancers, sorry, not the cancer cells, various type of the cells that they express different things. it goes and bind and then it gives you contrast property. The disadvantage, or FDA has been recalled this one, because although it has a good features, but shelf life of this material, the dextran, the coated dextran, after a while it has been detached and then nanoparticles are settling down. Okay, that's a reason, therefore, it's no more good. So another example of a formulation that we replaced, or it didn't do very well in the FDA approval process, that is bind 014 formulation. Constantly mentioning about Moderna formulation, they made liposomal for mRNA delivery. The same team, same investigator, they developed this formulation. So I mentioned PSMA is a good target for prostate cancer. Same, they have a targeting for the prostate cancer where that targets to the PSMA. But only thing was there was a over conjugation strategies they employed. But at the end, what happened? They choose docetaxel. They choose docetaxel PLGA formulation, this one, and also PLGA formulation, but that is conjugated with the PSMA. Okay, so these three, at the end, when they went for clinical trial, in the clinical phase 3 trial, they didn't find much difference between the docetaxel formulation and the docetaxel formulation with the targeting moiety. Okay, if you are unable to increase the amount of drug that is going to the tumors, okay, so means it's no use, right? So you make you choose a lot more complexation strategies and everything, but if we are unable to increase the drug amount that going to the tumor cells, there is no use, right? That's where they have failed. So similar, problems were with the formulation like CA-LLA-01. This was the cyclodextrin formulations, siRNA, and then again due to complex chemistry, and it didn't improve any drug accumulation at the tumor site compared to its plain drug injections. So with this, I want to stop here, and I want to get your feedback, and if you have any questions that we can discuss about formulations, how we develop, and we need to see the need of the formulation. Okay, if drug itself is doing better, for example, doxorubicin. Doxorubicin is a hydrophilic drug, right? You don't need basically any formulation. You can right away inject, right? That everybody agrees it doesn't require, right? But the problem was there, it's cardiac toxicity, okay? So we need to take care of the cardiac toxicity. Same way, whatever you take the drug, drug has a side effects or something, then we need to improve that particular aspect, okay? Here are some of the examples, but if you have any other drug that we want to discuss, we can discuss anytime.
Sir, this is a generic question regarding the Angelina effect. Since you are in US, the Angelina removed both her breasts, Angelina Jolie.
Mm-hmm.
Is there any impact on the society on sensitization of breast cancer prevention?
Yes, sir. So as I mentioned before, okay, especially for the breast cancer, at the age of 35, I think 40 is some age, every year women goes to annual check, okay? They undergo mammogram, they undergo if they see anything suspicious, and they do CT or MRI, okay? If they find anything, okay, they think that, removing the breast would be the easiest one. Okay, they don't do tiny mini surgery, because they do the surgery also, but the tumor size matters. Okay, if it is a bigger size, surgeon thinks that removing breast would be easier.
Sir, what about the longevity?
Longevity, I think it's good. It's good because removing breast is unlike other organs. Okay, so removing breast, it doesn't give you any other side effects. Okay, so anyway you can have some refilling, breast constructive surgery, all those are there. They will fill with the other materials like silicon based, other so you have it maintains its shape and everything, but that's the easiest one, that surgery is the easiest one. Then they do chemo and radiation, and I think that's a practice. Okay, so each surgeon or physician, they have a FDA approved guidelines. If somebody diagnosed with a particular cancer, they have a guideline, they follow. That's it. Okay, so they don't do, if any clinical trial going on different way, they try to do it, but those were like later on stage.
So could you shed some light on magnetic force?
Yeah. So for the, I was talked about Abraxane. Abraxane is the drug for breast cancer, okay, and lung cancer and pancreatic cancer.
Okay.
But you need to remember that whenever we talk about cancer, the primary management to the cancer is a surgery, if that can be operatable. But some cancers, by the time of detection, those are not operatable. Pancreatic cancer, it's very hard.
Very hard.
But by the time of diagnosis, pancreatic cancer, it's already those metastasized to the many other organs.
That was the case with Steve Jobs when he died in 2007.
Yes, Steve Jobs, he had already, stage 4 type. So he was treated in Memphis, where I was there in my earlier university. Okay, so he was treated in the Memphis Baptist hospital. Okay, so the thing is, it's same. Pancreatic cancer means the first one is they do Whipple surgery, means they take out the entire pancreas. Okay, but they do it if that is any beneficial. Okay, anyway you do the surgery or don't do the surgery, phase 4, okay, stage 4, patient, they don't live more than 6 months.
So he was at the 4th stage, Steve.
Yes, Steve Jobs, yeah.
4th.
Stage, yes.
Affecting these nanoparticles for inflammatory condition and degenerative conditions. Which way we can actually target? Because the inflammation is probably resolved.
Yeah, so inflammation is, so inflammation, if you talk about inflammation, so nanoparticles are good because they go to the inflammatory site. Okay, and I showed you one of the formulation that we coated different type of cell membranes, and one of the cell membrane, neutrophil decorated one, that goes to the inflammatory site. But you need to remember that majority of the cancers, they are associated with the inflammation. Okay, but if you talk about other than cancer, but still nanoparticles can be recruited at the site of inflammation. The Bayer formulation, it's inflammation, right? Which one? Like what type? Viral. See, that's what, like nanoparticles itself, it's not a therapy. It's a carrier, right? So you choose to come here from the hotel, you choose auto, rickshaw, whatever, by walk or car, bus, whatever way, it's a vehicle. So you need to choose which one is good for you or which one you can afford, right? So nanomaterial just giving you a medium that can overcome some of the associated drawbacks of that particular drug. Okay. Otherwise, it doesn't do. In some cases, nanoparticles itself act some way. For example, gold nanoparticles work as a sensitizer for radiation. Okay, and the other way, like some of these studies, they show that iron oxide nanoparticle, it improves the immunotherapy. Okay, PD-L1 immunotherapy, all those. Okay, but as such nanoparticle, they don't have much value itself. But when you are using the context of delivery, when you're using context of some other way, so it has a major value. Yeah.
And
Central nervous system like CNS delivery, right? CNS delivery, it's a major challenge, right? So if you give any drug, only not even 1% cross to the blood-brain barrier, right? So if you make any formulation that can take a little bit more amount of the drug, that is beneficial. So that's where we initiated some of the HIV projects, okay, that taking care of hand disease, like hand is a type of disease, like once person infected by HIV, that goes to the brain, and that condition we want to treat. So we have a oral pill of 3 different drugs, okay, but so that's oral pill. So our idea was like delivering the curcumin formulation and sensitize those cells in the HIV infected cells in the brain, sensitize to the oral drug. And one of our Ph.D. student is graduated with that work. And now, so I showed you one picture of like brain targeted delivery. So that was the one we are applying for Alzheimer's disease, okay, and Parkinson's disease, okay. And also like we have received another funding for the ALS, okay. So these are all like some of them are associated with the brain delivery. So that's where we propose that, but we have not evaluated its efficacy. That's the data showing effective delivery, okay. Always delivery is not going to turn out to be the drug activity, right? So that drug has to come out, drug has to do its function, right? So we need to evaluate that function. In India, okay, so I have no idea, but primarily what I know, the major FDA approved formulation, I said Doxil, right? Doxorubicin formulation. So you make any formulation, okay, you hold a patent rights up to a certain period of time, okay. After 20 years, anybody can make the same formulation. That's why we get what you call, there is a term for after expiration, generic, okay. So generic formulation, okay. So because like everybody knows that you can make doxorubicin liposomal formulation after 20 years, then the created company, okay, they lose the rights, okay. Other company can also make generic formulations of that particular formulation. Whatever we get mostly here in India, because of the cost effect, we get majority generic formulation. But some hospitals, some people can afford, that way we can use the other formulation also. But in my opinion, almost all formulation must be available in India also. The only thing, how much we can afford.
Some about the compulsory licensing of some cancer drugs in India.
So that's all like regulations. Like whenever you create a new molecule, they will have their own patent. They don't disclose as such until some time, until they get the patent. They don't disclose the actual structure of the molecule. Okay, once they get the patent rights and everything, and then that's why molecule may not be cost that much to prepare. So these are all going to add because they want to have the rights. So creating the molecule doesn't mean that to create that molecule it costs maybe $100. Okay, but to go that place, they need to do phase 1 clinical trial, phase 2 clinical trials, and phase 3 clinical trials. That costs a lot of money. Okay, so they cover once they have some patented and FDA approved formulation. They do 200 molecules, 200 patents, but only one formulation will turn out to be a FDA approved, right? So that means from that one formulation, they want to recover all 200 formulations they tried. They put money on it, right? It's non-profit. That's not non-profit organization. They want to get the money, right? So it's a business. Yeah.
Sir, actually we are using we are injecting a foreign material. So will there be any side effects due to this?
Foreign material, you mean drug or what it is? You mean nanomaterial? Yes. It's a good question. Whenever we use any material, that always we think we want to choose either biocompatible or biodegradable, right? So whatever we use gold nanoparticles, those are not biodegradable, right? But those are biocompatible, okay? And all nanomaterial that we inject, okay, it will be having a clearance mechanism. Some may come out from the body in a day, 2 days, or maybe 30 days, or even more. Okay, those are injections. Some of them are depots, right? So they do brain surgery and then they place one implant with the drug. Okay, so that stays for 6 months. Okay, so 6 months it stays and then it keep on releasing the drug. Okay, and in that time, we don't want to have a biodegradable quickly. We want to have only biocompatible and release the drug, right? So, that was we need to decide what application we are going to use and what type of material we want to use, okay? For any, bone reconstruction, all those knees replacement, all those are, we use titanium-based implants, right? So titanium-based implants those are biocompatible. Those are not at all biodegradable. It's a metal, right? So those are not biodegradable. So you need to choose what application we want to use, okay? So same, another example would be what I can give. Okay, so another example based on whenever we go to the physician, they do mammograms, right? If they suspect any cancer activity in the breast, what they do a fine needle biopsy, okay? So it's like they inject with the needle, and then they get some of the cells, okay, so that they don't do any bigger hole or anything. They get the cells and then they test it, okay. At that time, what they do, they replace the area where they got the cells or tissue. They replace tiny implant, okay. Tiny implant, those are titanium based, okay. They leave it there, okay, so that when the patient goes back, if the physician or the pathologist decide it's a cancer activity there, okay, then they can see where the cancer activity, then they do the radiation, okay, because that pin or that implant give a guidance to the physician where we put the cells, and then they do the radiation therapy, okay. Or the same way they pick like multiple areas, okay, where they have a concern, okay. So they do the radiation, okay. If it's a bigger tumor, then eventually they do the surgery.
Sir, what about stents where they are biocompatible or biodegradable?
Stent is biocompatible. Stent is it's a metal basically. It's a metal. Yeah. It's not degradable. If stent degrades, then it's see how again the cells will grow, and then you see the blockage of the heart valve.